wondering. Good to be back for the second part of this first day of the webinar of the European Cetacean Society 2021. We have our first presenter, Heidi Pearson from the Department of Natural Sciences of the University of Alaska Southeast. But before I give the word to her, I like to stipulate again a few instructions for part of the audience which just have arrived at the webinar. The speaker in this case is Heidi Pearson. We'll have about 30 minutes for her talk, and about 5 minutes before she should be closing her presentation, she will hear a bell, a soft bell, which will tell her politely, I hope, that she has a few more minutes to round up her presentation. After the talk, there is again a minute to like the written questions, which you can add as well. You will find at your Zoom bar on the button question and answer. If you push or click on it, you can write a question. At the end of the talk, you can go back there or during the talk and like a question or not. And the more likes a question gets, the higher it will appear on the list. And I'll start reading out the questions after the talk. And I'll start with the one which is the highest. And of course, the presenter will start answering the questions one by one, not in one sequence. So again, good morning, Heidi. I believe it is morning at your side of the earth. You're in Alaska. Looks good there on the background. You will present us, well, we can already see it on the screen, Cetaceans as Oceanic Engineers. It's the second talk today about the interaction of climate change and megafauna in our oceans. So I'd like to give the word to you, Heidi. So please take the virtual floor.
Thanks, Frank, for that great introduction. And thanks to the ECS organizing committee for inviting me to speak to you. And also thanks to all of you for joining. Well, so I'll start with a few photos here from my backyard in Juneau and observations of this glacial retreat since I moved here about 10 years ago really got me thinking even more about climate change and the marine environment and what might be done about it. So this is Mendenhall Glacier, and there are 39 glaciers that extend from the Juneau ice field, which is the border between Juneau and Canada. And Alaska and British Columbia's glaciers are shrinking at one of the fastest rates on Earth. And we can clearly see that here in these photos. So the top two are showing the retreat of this glacier from 1993 to 2010. And then the bottom two are my photos from shortly after I moved here to just last week. And what's really apparent to me anyway is what I call a shrinking of the waist of this glacier. So you can see especially these rock ledges are becoming more and more exposed on either side of this glacier. So of course, the reason for glacial retreats and climate change is rising atmospheric carbon dioxide levels. So this graph shows the increase in CO2 levels since they were first started being measured on Mauna Loa in 1958. So our current level is shown here. It's about 417 parts per million. But alone, this number might not be that meaningful. So I think it's useful to put it in a larger context. So let's look at some other benchmarks of atmospheric CO2 over the past few decades. So in 1988, Earth reached 350 parts per million, which is considered the tipping level, at which point we start to see large-scale climate impacts occurring. Then in 2013, CO2 levels exceeded 400 parts per million for the first time in approximately 2 to 15 million years. And this is important because our species, Homo sapiens, has never before existed on the Earth in CO2, in a time when CO2 levels are this high. And so the final benchmark here is 450 parts per million, which is a level at which very large scale climate impacts are expected to occur. And this has also been called the point of no return, at which point the Earth will enter an ice-free state. And some projections indicate that if we continue on the path we are now, we could reach this point in 2040. So there are, of course, many strategies for reducing atmospheric carbon dioxide emissions and other greenhouse gases. One natural strategy is what's called blue carbon, which refers to atmospheric carbon that is naturally trapped by marine life. So the oceans absorb about a quarter of all anthropogenic carbon dioxide emissions, and this is an ecosystem service. So something the marine environment naturally does that benefits humans. And to me, blue carbon has always made a lot of sense because it's just another reason to protect the marine environment. So not only does marine conservation lead to higher biodiversity and healthy ecosystems, but it might also be able to aid in climate change mitigation. So blue carbon traditionally refers to coastal vegetation ecosystems like seagrass beds, mangrove forests, and salt marshes, but it's been more recently applied to marine vertebrates. So this figure shows 9 ways in which marine vertebrates can aid in nutrient cycling and carbon storage and sequestration. The role of cetaceans has been quantified in 4 of these mechanisms, and those are the ones I'll be focusing on today. And they are highlighted here in yellow. So it's the whale pump, the great whale conveyor belt, biomass carbon, and deadfall carbon. To learn more about the rest of the mechanisms, I have a link to our story map at the bottom left that you're welcome to visit. So just briefly here, the whale pump and the great whale conveyor belt are ways in which cetaceans release nutrients that can stimulate phytoplankton growth. So this not only enhances ecosystem functioning, but it can also enhance carbon storage and sequestration by phytoplankton. Through biomass carbon, cetaceans can store large amounts of carbon for long periods of time due to their large body sizes and long lifespans. And then when they die, they oftentimes sink to the seafloor, and they can take that lifetime of trapped carbon with them. The final thing I want to draw your attention to on this figure are the 100 and 1,000 meter depths. You'll notice that some of these mechanisms occur towards the surface, while others occur more deeply. And all of this has implications for the carbon functions of these mechanisms. This is a figure of the biological carbon pump, and here we can take a closer look at what those depths mean in terms of carbon. So once carbon reaches a depth of about 100 meters, it is considered stored and out of the atmosphere on a timescale of decades. Once carbon reaches a depth of 1,000 meters, it is considered sequestered, and here it can remain out of the atmosphere on timescales of thousands of years. And then if carbon reaches the deep seafloor, it can become buried in sediments and remain out of the atmosphere for potentially millions of years. I'll also draw your attention to the allochthonous nutrient input at the top of the graph. And this refers to new nutrients that enter surface waters from outside the surface layer. This is either through terrestrial inputs like rivers or processes that bring nutrients up from depth, like upwelling, or these processes through cetaceans that I'll go through. And in terms of carbon flux, this new nutrient input is considered quantitatively equivalent to carbon export or sequestration to the deep ocean. In contrast, autochthonous nutrients refer to nutrients recycled within the surface layer, which can aid in carbon storage. Through their natural actions, cetaceans can change their biotic and abiotic environments. So this fits the definition of an ecosystem engineer. When you think of this term, you might think of other organisms such as beavers, prairie dogs, earthworms, elephants, sea otters, and coral reefs. However, in this talk, I'll focus on how cetaceans are also ecosystem engineers. The role of whales in providing ecosystem services is gaining traction. And at the last meeting of the IWC, a total of 41 countries endorsed resolutions that recognize the ecological role of cetaceans in biodiversity, carbon cycling, and climate change. So the first two ecosystem roles of cetaceans I'll describe are actually all about poo. So whale poo comes in all different shapes and sizes and colors, but the most exciting thing about it is that it floats, and whales produce a lot of poo through their natural digestive processes. So due to the dive response, cetaceans slow down their non-vital functions, including digestion, while diving, and then they tend to defecate when they are recovering and resting at the surface. So these fecal plumes released at the surface contain nutrients that are important for phytoplankton growth. And this is important not only for forming the base of the food web, but it also aids in carbon storage and sequestration. So through the whale pump, whales release nutrients in surface waters from prey obtained at depth. Depending on the depth of the prey then, these nutrients can be considered new or recycled. Through the great whale conveyor belt, migrating whales transport nutrients from high latitude feeding grounds to low latitude breeding grounds. These nutrients can come in the form of poo, but also urea and placentas. These nutrients then are considered new because they originated on the feeding grounds, but are released thousands of miles away on the breeding grounds. So whale poo isn't always so easy to spot. So see if you can spot the fecal plumes in each of these photos of humpback whales. So hopefully that wasn't too hard for you. There's 1 and 2 and 3. So again, the reason that this whale poo is exciting is because it contains nutrients. So these nutrients are considered limiting nutrients because they are typically present in the surface waters of the ocean in small amounts. But when there is an influx of these limiting nutrients, it essentially acts like a fertilizer and can stimulate phytoplankton growth. So the primary limiting nutrients that I'll be talking about here are iron, nitrogen, and phosphorus. So there have been several studies that have looked at the limiting nutrients present in fecal plumes and their impact on the ecosystem. So one study from the Southern Ocean found that fecal plumes from sperm whales were found to have iron levels that were 10 million times higher than background seawater levels. All of this iron was then estimated to stimulate new phytoplankton growth and carbon sequestration. The graph here shows the estimated amount of carbon exported to the deep sea and also respired by sperm whales from 1650 to 2005. And you also see that upward line there, which is the amount of carbon emitted from fossil fuels. The amount of carbon export stimulated by a modern day population of 12,000 sperm whales was estimated at 400,000 tons. But as you can see, as we go to the left in the graph, earlier in time before whaling, the amount of carbon exported by sperm whales would have been orders of magnitude larger. Fecal plumes from humpback whales in the Gulf of Maine were found to have nitrogen levels that were 550 times greater than in seawater. These graphs on the right show the nitrogen flux attributed to whales and other sources in the present day on top and pre-whaling on the bottom. So the top graph shows that the nitrogen input to the Gulf of Maine from whales was greater than the nitrogen input from all rivers combined. The bottom graph shows that, similar to the sperm whales on the previous slide, this nitrogen input would have been orders of magnitude larger prior to commercial whaling. When whales migrate across the globe, they transport limiting nutrients. Here, we see the distribution of gray, humpback, and right whales on their foraging and calving grounds. Low latitude calving areas are typically nutrient poor, and so this influx of new nutrients can have a large impact. One study estimated that a pre-whaling population of blue whales in the southern hemisphere could have transported enough nitrogen to stimulate 130 million tons of carbon sequestration annually on low latitude breeding grounds. In the North Pacific, where I work, most humpback whales migrate between Hawaiian breeding grounds and various high latitude feeding grounds in British Columbia and Alaska. These whales are part of the Hawaiian distinct population segment, and this population segment is estimated to contain 20,000 individuals. This population segment is growing and is not considered to be endangered or threatened. And 96% of the humpback whales that occur in Southeast Alaska, where I work, are also part of this Hawaiian distinct population segment. The recovery of this population segment is really a nice example of a conservation success story. So after being decimated by commercial whaling, humpback whales received protection in the US through the Endangered Species Act, the Marine Mammal Protection Act, and then the moratorium on commercial whaling by the International Whaling Commission. Since then, the population has rebounded and is currently increasing at about 5 to 7% a year. In northern Southeast Alaska, where Juneau is located, over 1,500 humpback whales return each summer. This is my study site near Juneau, which is in the panhandle of Alaska. This is also right along the Inside Passage. So to give you some perspective, Seattle is about right here, and then we're about 900 miles north. And zooming into my field area, here's downtown Juneau, here's Auk Bay, where the University of Alaska Southeast is, and my study area is all through here. And we reliably see humpback whales from April through about the end of September. So this is a great area to study the whale pump because the whales are here during the summer. Their main goal is to feed and put on all those blubber reserves that they lost while fasting on the feeding ground. And also no other studies so far have looked at the whale pump in the North Pacific. So for the past few years, my team and I have been studying aspects of the whale pump here in Southeast Alaska. And overall, this is a very dynamic place to study the whale pump because glacial melting may actually be increasing nutrient input into the coastal ecosystem in the short term. And this can then have bottom-up ripple effects on phytoplankton, but also many other parts of the food web, such as salmon, which are both ecologically and economically important here. So through the whale pump, the question is, do humpback whales release limiting nutrients in their fecal plumes, which can then further enrich coastal coastal waters? And can this increase in nutrients then support more fish? And can more fish then help to support more whales? So this is the big picture question, and we're currently working to break down individual pieces of this larger question. So as I mentioned, humpback whales come to this part of the world in the summer to feed, and feeding, foraging is their main activity. Feeding can occur in several different ways. So it can occur below the surface. And this picture on the left is an echo sounder snapshot where we see a depth of about 127 feet. We see the prey layer between about 70 to 100 feet. This prey layer happened to be herring. And then above that, you see that arched shape, which is actually a humpback whale going down to dive. So humpback whales can feed below the surface. They can also feed at the surface as well. So this middle figure shows a single humpback whale lunge feeding at the surface. And then the bottom right shows the spectacular bubble net feeding behavior, which occurs in Southeast Alaska, which can be done in cooperative groups, and it can also be done alone. And not all individuals in Southeast Alaska bubble net feed, and it's thought to be a cultural tradition because it tends to be that only certain individuals will bubble net feed. But no matter how they feed, humpback whales eat a lot. They eat about 1 ton of food per day. And in Southeast Alaska, their primary prey are not only herring, but they'll also eat capelin, krill, and smaller salmon. So the first thing we did to assess the whale pump was to measure phosphate levels, so one of those limiting nutrients. and humpback whale feces, in addition to individuals that we got from necropsies. So we also analyzed feces from a minke whale, a fin whale, and a harbor porpoise. We dried these samples and then diluted them and ran them through a nutrient autoanalyzer to assess the nutrient levels. And then we also ran our seawater samples through this autoanalyzer as well to get the baseline level of nutrients in the ecosystem. So compared to the seawater nutrient minimum, phosphate levels in humpback whale feces were 705 times higher. Looking at minke whales, those phosphate levels were 665 times higher. Harbor porpoise showed 682 times higher levels of phosphate compared to seawater. And then finally, fin whale showed 823 times higher phosphate levels. So taken together, the average is 715. So on average, these cetaceans had phosphate levels in their feces that were 715 times higher than the background seawater levels. So this indicates that there is great potential for these animals to contribute nutrients to surface waters. So the next thing we did was to run an ecosystem model using qualitative network modeling. So we wanted to examine not only the role of whales in nutrient cycling in the ecosystem, but also the other top predators in the area, which are pinnipeds and seabirds. So this type of modeling, this qualitative network modeling, predicts how an ecosystem responds to a perturbation or some kind of disturbance or change. So this figure shows our simplified model of the Southeast Alaska food web. Based on the existing literature, we identified the node of each linkage as being positive, which is shown by an arrow, negative, shown by a circle, or neutral, shown by just a line. So for example, the link between zooplankton and baleen whales is negative for zooplankton and positive for baleen whales. We can also look at the link between adult chum and pink salmon and pinnipeds, and we see it's positive for pinnipeds and negative for the salmon. This is obviously a simplistic view of an ecosystem, but it is useful as a starting point, particularly in data-limited systems like Southeast Alaska. So we then ran 8 models to determine how the ecosystem would respond to various scenarios. So we increased baleen whales, pinnipeds, and seabirds individually and then together. And then we increased nutrients, primary production, zooplankton individually and then together. So this graph is showing the various models. So the things that we changed in the ecosystem on the x-axis, and then the response of each item within the ecosystem on the y-axis. And the colors in those boxes represent the response. So the bluish colors represent a positive response, and the reddish colors represent a negative response. And then the intensity of the color corresponds to the intensity of the response. And then finally, those X's represent a neutral response. So I want to highlight 2 main things that we found with our models. So first of all, increasing baleen whales alone, which is the leftmost column, did not increase nutrients. But when we increased baleen whales along with pinnipeds and seabirds, we found an increase in nutrients and primary production. So this indicates that there may be some sort of synergistic reaction between these top predators that's necessary for nutrients to increase in this ecosystem. Second, we found that increasing nutrients had the most positive effect throughout the ecosystem of any model that we run, speaking to the importance of nutrients as the foundation of marine food webs. So this is a starting point for this research, and we're currently working on proposals to further break down these models and specifically to get empirical data to do more quantitative modeling. Most of my work has focused on the nutrient cycling functions of cetaceans, but I wanted to come back to this figure to go through 2 other mechanisms in which cetaceans are involved, and these are biomass carbon and deadfall carbon. Because whales are large, such as blue whales, which may weigh up to 150 tons, And they're also long-lived, such as bowhead whales, which may live for over 200 years. They have the potential to store large amounts of carbon for long periods of time. So you can think of this as being analogous to the carbon stored in large, long-lived trees. In terms of biomass carbon, bigger is better. Large animals have a high metabolic efficiency, and the same amount of food can support 1 blue whale, 7 minke whales, or 1,800 Adelie penguins, and this is shown in the top graph. However, the total biomass that this amount of food supports is 92 tons for the blue whale, 49 tons for the minke whale, and 9 tons for the penguins, and this is shown on the bottom graph. So that extra carbon that originated in the prey would then go to the atmosphere. So essentially, in terms of carbon storage, bigger animals are better at keeping carbon out of the atmosphere. The final mechanism I'll mention is deadfall carbon. So when a cetacean dies at sea, as this figure on the top left shows, it has many different pathways that it can take. So the carcass can float for a time and then wash ashore. It can float and then sink. It can float, sink for a time, then refloat and either wash ashore or sink again, or that carcass can sink and remain at depth. And it's likely that most carcasses do sink. And in particular, once a carcass reaches about 100 meters, it is predicted to sink to depth and remain there. When carcasses sink, they take with them that lifetime of stored carbon that can then be sequestered for hundreds of years or more. In addition, these carcasses create rich, diverse ecosystems in regions of the deep sea where food is typically scarce. The figure on the bottom shows the decomposition of a gray whale carcass and the organisms feeding on it, such as hagfish, bivalves, polychaetes, crustaceans, gastropods, and bacteria. So going back to these figures, I wanted to talk about differences in the amount of carbon stored and sequestered between pre-whaling or pre-exploited populations and more modern day populations. So in terms of the amount of carbon stored and sequestered, whaling and bycatch have not only decimated populations, but also have decimated these natural carbon stores. At the top of each of these figures is the pre-exploitation carbon value, and at the bottom is the post-exploitation value. So let's start with the whale pump. In the Southern Ocean, commercial whaling led to an estimated 83% decline in the amount of carbon sequestered by sperm whales through the whale pump, and also an estimated 98% decline in the amount of carbon stored by blue whales through the whale pump. In terms of the great whale conveyor belt, commercial whaling reduced the amount of carbon sequestered by blue whales by an estimated 99%. Continuing on, in the Eastern Tropical Pacific, bycatch resulted in an estimated 85% reduction in the amount of carbon stored by spotted and spinner dolphins. And finally, commercial whaling led to an estimated 66% reduction in the amount of carbon stored by 8 baleen whale species globally, and an estimated 85% reduction in the amount of carbon sequestered through deadfall carbon by these species. So that was a lot of numbers. So to summarize, there has been an estimated 96% reduction in the amount of carbon stored or sequestered due to exploitation of these cetaceans. And to put this in perspective, this is analogous to losing the carbon sequestration ability of approximately 288 million hectares of forest annually, and that's about the size of Argentina. However, this is a very broad preliminary estimate, and we still have lots to learn in terms of the role of cetaceans in nutrient and carbon cycling. It's important to remember that only a handful of studies on a handful of species in very few areas have provided quantitative estimates. So we have a great starting point, but we still have a long ways to go to support and refine these numbers. In particular, there are many unknowns regarding if and how cetaceans stimulate phytoplankton growth and carbon sequestration. This is a conceptual diagram on the contribution of marine organisms to iron cycling in the Southern Ocean, but we can apply these same principles to other regions. My lab is currently working to understand if the foraging dive depth of humpback whales occurs above or below the mixed layer, which will determine if those nutrients from the fecal plumes are new or recycled, which has implications for if that carbon can be stored or sequestered. We also don't know what proportion of whale-derived nutrients are actually taken up by phytoplankton or that enter the microbial loop and are taken up by bacteria and viruses. We plan to do incubation experiments to test this. And then finally, if whale-derived nutrients do stimulate phytoplankton production, we don't know the proportion of that carbon that actually sinks to depth versus that which enters the food web. So the good news, though, is that the data needed to fill in many of these knowledge gaps might be collected as part of existing studies. So for example, data needed to further test the whale pump might already be part of the standard operating procedure in tagging studies. Data needed to test the great whale conveyor belt could be collected opportunistically as part of ongoing work on the breeding grounds. And finally, estimates derived from ongoing studies of population dynamics could be incorporated into models for biomass and deadfall carbon. So I'd love to discuss any of this further with you. If you're interested, please feel free to email me. In closing, this is a huge topic, and while intriguing, we still know relatively little. But through collective action, I think we can get closer to further understanding the role of cetaceans in nutrient and carbon cycling. It's also important to remember that conservation of cetaceans is just one of many potential strategies for mitigating climate change. We have a lot of work to do and to do it quickly so that we don't pass that 450 part per million threshold. And certainly strategies that directly reduce greenhouse gas emissions should be used alongside strategies to promote marine conservation. It's also important to be mindful that cetaceans are inherently vital components of healthy ecosystems and that they have many other intrinsic values. The work I discussed throughout this talk was built on a strong foundation of work by others, which I've cited throughout the talk, and I'm very grateful for that work. I've also been fortunate to work with a stellar team to assess the ecosystem role of whales here in Alaska, and we've been supported by various funding agencies. So with that, I'd like to thank you for your time and attention, and I'm happy to answer any questions that you have. Thank you.
Okay, thank you, Heidi. This was very, very interesting. And I hope and I think it was very interesting for the audience as well. But they probably with some questions, and I will give them a minute to like others' questions or to write a few more. So I'll get back to you and the audience in a minute time. Okay, this should be something like a minute, and it won't stop. You can still like the questions, but a silent gap for a long time is not really a thing that we like. Let's see what is the most liked questions. This is Simon. Heidi, how much of these nutrients really move along their migration route, given that feeding is limited, if at all, and they must evacuate, it's going up and down, their intestines quite quickly? Does most nutrients are left on the feeding grounds?
Thanks for that question, Simon. And this speaks to some of the unknowns. And so the mechanism that I believe you're talking about here is the great whale conveyor belt. And, you know, whales, I actually haven't seen it personally, but I have heard from colleagues that whales do defecate on the breeding grounds. So it can happen, but we think that most of the nutrients deposited on the breeding grounds are from urea in urine, which comes with its own challenges in collecting that, but also through byproducts of birth, like placentas, like I mentioned. And so we, this is one of those unknowns. Initially, it might be that the nutrient deposition on the feeding grounds is higher, but also remember that these breeding grounds in low latitudes are typically very nutrient poor. And so I think there could be a larger relative impact for this influx of nutrients on the breeding grounds than the feeding grounds, because feeding grounds are inherently more nutrient rich. Oh, Frank, you're muted.
Sorry, I muted myself. We proceed to the second question. There's Kevin. He works with minke whales in northeast Scotland and would be interested to collect fecal blooms. But in reality, how easy and practically can this be done?
It really varies on the species, and even within the species, it can vary on what they're eating. So here in Southeast Alaska, when the humpback whales are feeding on fish, that tends to make the fecal matter heavier and it tends to sink more rapidly. But when they're feeding on krill, the fecal plumes tend to be more buoyant. So it depends on what they're feeding. And the other thing is that you just have to always be watching, I guess I would say. And typically when humpback whales do release a fecal plume, and again, this can vary by species, but it oftentimes occurs right before they go down for a dive. So one strategy is after they go down for a dive, you can go over their fluke print and see if you can see any floating fecal matter. Another technique that I hope my team will be able to use pretty soon is to use a drone. I know other colleagues in Southeast Alaska with the Alaska Whale Foundation, for example, are having success with just part of their ongoing drone work, also looking for fecal plumes, and you can spot it much more easily from the air. And then if you have a boat on the ground, you can hopefully zoom in and get that fecal material before it sinks.
Okay, thank you. I think this is practically handy for one who likes to go out on the water and collect some fecal plumes as well. The third question from Amy Myro. I was wondering, how is it possible that harbor porpoises show show a higher nutrient level compared to a much larger minke whale? Is this diet dependent on of just coincidence because it was only one animal?
I think it could be all of those things, Amy, and these are preliminary data. I think, relatively speaking, all of those animals that I showed on that slide it was about 700 or so times higher. And it can be because, we had an N of 1 for that harbor porpoise. It could potentially vary by diet as well, but that is certainly something to further explore to see if there is a size dependent effect. But my initial hunch is that I don't think that actual size of the animal will affect it, because it's a nutrient concentration which is already scaled to mass of the material. But it'd be something further to investigate for sure. Thanks.
Okay, thank you, Heidi. Since there is a question from Jesus, since there is this need to dehydrate fecal samples for the nutrient analysis, I wonder how much sample do you need to collect at sea for each analysis? Do you have a minimum established or a mean in liters, for example?
You don't need very much at all, really. Our protocol states that we collect, if we can, a liter, but that is a liter of the water and the fecal material floating in it. But to actually dry it and then run it through the auto analyzer, you need pretty small samples. So I think really anything you could get would be useful.
Okay, thank you. I think it's a clear answer. Then there's a next one from Rob Fujita. Are the enabling conditions for large increases in cetacean populations present, so that if we were able to dramatically reduce cetacean mortality, these populations would increase enough to have a significant effect on carbon sequestration?
Yeah, that's a great question, Rod, and something that I think about all the time. So the key word here in your question is significant effect. You know, we know it has some effect, but one of the amounts that I've heard just very offhand is that we might need to be looking at effects on the scale of gigatons, which we clearly aren't there yet with cetaceans. But you know, if we were to, as you say, dramatically reduce cetacean mortality, and we get all these benefits through the biomass carbon storage and the nutrient influx and so forth, I think certainly it would have an effect. But how much, we aren't there yet to answer that question. But certainly it would be more than we have now.
Okay, thank you. Then there's the question from Jay Barlow. How does the carbon in all pre-exploitation whale populations quantitatively compare to the accumulated human input of carbon to the atmosphere?
The input of carbon in the atmosphere is clearly more. And if you remember back to that graph I showed from, it was Trish Lavery's paper from 2010, where she showed the amount of carbon exported to the deep sea by sperm whales, and it was flatlined. And then, you know, post whaling, it took a deep dive down. And then on that same graph, you just see carbon emissions from fossil fuels going up like this. So we still have the main strategy, as I said in my talk, is still to get the human emissions down. But if we can get those down and also bring the cetaceans up, I think all of that together can work synergistically.
Thank you. There's a question from Astrid Fuchs. A lot of the studies, if not most to date, have been focusing on large whale species. What small cetacean species would you recommend focusing on? Would deep diving species like big whales or pilot whales be particularly interesting?
Yeah, certainly, Astrid. That's a great question. And I think it would be really exciting to study this in beaked whales because, of course, they are some of the deepest diving marine mammals on the planet, if not the deepest diving. And so they certainly would have a huge impact on this influx of new nutrients in surface layers. And the trick then is to get those fecal plumes, or, you know, a good starting point for those species that might be hard to collect data on in the field is to make sure that when we have necropsies, that we get some feces from the animal and then analyze that. And I think that's another great area to advance, or great way to advance this area of research, is through stranding networks. You know, you have an animal on the beach, it's just very simple, hopefully, to cut open the colon and grab some feces and archive it, and then you can always freeze it and then analyze it later. But certainly, Astrid, I think there's a lot of other interesting things that will come out from these non-baleen whale cetaceans.
Okay, thank you, Heidi. Proceed to a few more questions. One from Jack Sheehy. Do you have any thoughts on how could you quantify the ratio of whale falls that make it to the seabed, which could then be defined as sequestered carbon?
Sure. And Andy Pershing has done a lot of work with this modeling, and I cited his 2010 paper. And I believe in that paper, he used an estimate of 50% of whales that died go to the deep sea and then essentially are there forever. So the assumption is that once these carcasses, you know, they're big and heavy and they sink, and they're just going to stay. When they rest on the deep sea floor, they're going to stay there. And then through the decomposition processes and all the rest, they will stay there, and their carcasses will get embedded in the deep sea sediments. And so the assumption is that that carbon then enters the sediments in the deep sea and can be there for millions of years in some estimations.
Okay, thank you. Then a last question. What's the difference between nitrogen pollution from rivers coming into the sea in comparison to nitrogen coming from the whales?
Mm-hmm. Great question. And I think here you might be referring to excess nitrogen, which might cause dead zones or harmful algal blooms. And there's a couple of differences. So one is that the whales, of course, will be releasing these nutrients in a more natural way. It's not going to be such a huge, pointed impact at one time, like we see. I'm thinking of the Mississippi River dead zone in the Gulf of Mexico, for example. And the other difference, I think, has to do with the type of phytoplankton species that are present that are actually going to be taking up those nutrients.
Okay, thank you. You meant by your answer that the concentration from rivers is higher?
Well, if you're talking about large influxes if you're talking about the negative impact of nutrients from water, such as say the Mississippi, where there's a lot of agricultural runoff and nitrogen, that certainly is higher than the whales. But if you remember back to that figure I showed from Joe Roman's 2010 paper, just from natural input of rivers, they found that whales collectively had a larger nitrogen influx. So I think that question, you need to think of it in terms of the scale, the spatial scale and the temporal scale of the nutrients.
Okay, thank you. Oh, I thought there were some questions left, but they disappeared.
Oh.
They are withdrawn, or I don't know. But I don't see anybody typing, so I think we have come up to the last question. So thank you, Heidi, for your very clear presentation. As you know, it's also a presentation of my interest, but I even hope, well, you raised a lot of research questions at the end, but it's still not known that it might well, have an effect on all the researchers still in the audience thinking about new topics and what could even help the world. Okay, there's one more question. I think we have time for that. As you said in the beginning, restoration of marine ecosystems, mainly marine seagrasses, is an alternative to land reforestation. Thus, which will be the better way to engage MPA managers to sell and give value to carbon compensation in terms of this blue carbon linked to big size cetaceans?
So I think your question is protection of cetaceans better or worse than these coastal vegetation ecosystems?
I think that is meant by the question, yes. Yeah.
Okay. And well, my ideal answer is we should do both because they certainly both will help. But I know that the science is certainly further along in understanding carbon flux within these coastal vegetation systems, seagrass beds, mangroves, salt marshes, than it is with cetaceans and other marine vertebrates. And in fact, as part of some countries' nationally determined contributions, NDC, to the Paris Climate Accord, some of them have already identified protection, restoration of these coastal vegetation ecosystems as part of their contributions to reducing their own country's climate or their carbon emissions. And so I know that just based on that alone, that is certainly better understood at the moment, but there could be even greater potential for cetaceans and other large marine vertebrates to contribute. So I think that as we keep moving the science along, we can be in a better place to start making policy that involves cetaceans and carbon as well.
Okay, thank you, Heidi, and I also like to thank Camille for both of your presentations. Both were on climate and whales, and I also like to thank the audience, which showed up in large numbers. So thank you, and also for the questions you all raised and have written in the boxes. We as ECS Council hope that you enjoyed and learned from this first day of the webinar. And we are looking forward to meet you again tomorrow, not in the afternoon, but we start with the second day in the morning. Please have a look at the ECS website, where the program will you tell more about the specific presentations. So again, thank you all for being here, sharing your thoughts, questions, and your knowledge, and hope to see you soon again. Bye-bye.